Hello, welcome to the first in our series of AFM webinars. My name is Paul West, and I'll be making a presentation today. And today's topic is an introduction to AFM image processing. During this presentation, we'll be talking a lot about Gwyddion software, which is a free software package that you can download from the internet. Gwyddion is a software package that comes with our TT-AFM, which I have right here next to me. The TT-AFM is a product which is designed for nanotechnology research, education, and instrument innovators. So this is an introductory talk, and this is meant for people that are just starting to learn about atomic force microscopes and need to learn about image processing. The talk will last about 20 minutes, so it won't take up too much of your time. The topics we will discuss are a brief introduction to image processing for AFMs. Then we will talk about processing images. We'll talk about displaying images, and we'll talk about analyzing images. These are the three primary functions which are used in image processing software for atomic force microscopes. The first thing I'd like to say is AFM is very similar to other microscopy techniques, that is, in terms of the image processing. There are two things that are very different. The first one is that atomic force microscope images are three-dimensional in nature. That is, you get X data, Y data, and Z data. And so therefore you can make three-dimensional topography plots which have correct depth information. Second is there's an explicit use of multiple channels, and that is that you can get two channels, say the topography image and the phase image, or the topography image and the lateral force image. Okay, so the first thing is make sure to back up your data. It costs a lot to buy one of these instruments, and it costs quite a bit to measure the images. So as soon as you get images, back them up on a memory stick or on a hard drive. Make sure you don't lose your data. All AFM images do need some kind of image processing. It's very rarely that you would measure an AFM image that you wouldn't have the data process to get a good image. Thirdly, it takes longer to measure images than to process them. So it may take a minute to 10 minutes to measure an image. You can usually process an image in a few minutes, even sometimes a few seconds. Image processing allows visualization of hidden features in the AFM image. So when you measure an image, quite often you don't see a lot of features, but through the image processing sequence of events, you can start to see hidden features. And finally, when you do your image processing, you need to keep in mind if you're doing it to visualize the surface, that is to make a pretty picture, or to measure surface features. These have a different standard in terms of the integrity of the data and how you treat it. So, as an introduction to the software options, there are two open source packages that we're aware of. The first one is Gwyddion, which is available on the website. If you Google Gwyddion and AFM, you can get that. And the second is ImageJ, the Fiji implementation. You can also download that from the internet. Both of these are open source and free, and they're fairly full-featured image processing software packages. There's also commercial packages, which would be SPIP and Mountain Maps. And these commercial packages have more features than the open source packages and allow you to do many more types of image processing steps. Today we're going to be talking about Gwyddion, which is shown right here, and this is the interface. And it has the three primary functions, that's processing data, displaying the data, and analyzing the data. So an AFM image, when it comes into the computer, is a three-dimensional array of numbers that represent a sample surface. So it's got X, Y, and Z. When you process the image, you change the numbers in the array. That is, you take the numbers and you modify them. When you display the image, you simply change how the numbers are viewed. That is, the numbers don't change, it's just the representation of the numbers. It could be a color or a type of plot. When you analyze the data, you extract numbers from the data array, and so that's what's used for doing analytical analysis. So today, I'm going to talk about the first topic here that's in the red box. That's the leveling functions. There are other functions for processing, filtering and deglitching. You can learn about those from the Gwyddion website. The first reason you have to level images is to remove tilt. And I've shown right here a picture of a tip scanning up and down a sample surface. And if you look at this, you'll see that quite often the features you're looking at are even smaller than the tilt on the surface. So what happens then is when the image comes into the microscope, it'll be dark on one side and light on the other, and that's shown here on this test pattern. And quite often, the tilt is much larger than even the features, so you can't see the features. So the first thing you have to do is remove this tilt. Second, when you use an atomic force microscope, quite often you'll see the streaking in the image, which comes from many sources. It could be vibrations, it could be thermal drift, or it could be that the tip picked up a small nanoscopic piece of material on its end, and it changed the height of the image. And so you have to remove these line-by-line variations. So the most common leveling techniques are a surface subtract, a line-by-line subtract, and a three-point subtract. So I'll talk about these now. In surface subtraction, what you do is you use a mathematical equation, shown here, to model or to fit an average surface to the image. And then you subtract that average surface from the image. And that's shown right here. In Gwyddion software, there's an icon that you can click. And if you take this image and you click that icon, you'll see you get this image. And this image does not show this height variation across the length of the image from the top to the bottom. And then all of a sudden you can see more detail in the image. For example, you can see this contamination spot and the details of these little spots. And this is simply done by pushing this button, and it's a plane level. You can also do polynomial leveling with Gwyddion software and most software packages. That is by clicking on Data Process, Level, and Polynomial Background. You'll get this screen here, which allows you to change the order of leveling, shown right here. So this shows the plane or the surface that will be subtracted, and this shows the images how it will look after subtracted. Line-by-line leveling is used, usually in most AFMs, it's used for real-time display. So when you're running your microscope and you see a leveled image, it's because you're doing line-by-line leveling. And the way this works is it takes a line and it calculates the average, when I say line, I mean a line of the image, and it fits a least squares line to that and then subtracts it. And so it would do the first line, and then it would do the next line. And then what it does is it sets the average of the second line equal to the average of the first line. And so this is done, again, in real-time display. And it does create some artifacts that I'll talk about in a few minutes. So this is an example right here. This is this image of nanoparticles. And you can see that if you take the Gwyddion software and push this icon right here on this image, it removes those lines and it levels the image out, so now you can see the nanoparticles quite nicely. There are still a few little lines left in here that you can see, but it takes care of most of that unwanted image artifact that occurs because of the line-to-line variances. So you can create some artifacts and images by doing line-by-line leveling. This is an example right here of the test pattern. And if you do line-by-line leveling, it introduces these bands in the image. And those are artifacts of the leveling technique. However, when you do the plane leveling, you don't see that. Now, the point here is that you have to choose the correct type of leveling for the image. That's why you need to know something about these different techniques. This is a very common artifact that's introduced into images when you do line-by-line leveling. This is an image here of E. coli. And when you do line-to-line leveling, it puts these bands into the image. Now, on Gwyddion, you can do masking. And that's this icon right here, and allows you to draw a box around the different features on the surface, and then allows you to exclude those when you do the line-by-line leveling. And you get an image like this, which shows the E. coli without this banding in the image. So I'd like to stop now and ask, does anybody have any questions they'd like to ask on the chat? If you do, go ahead and ask the questions, and then I'll try to answer them. So my assistant is telling me we don't have any questions, so we'll just move on to the next subject, which is three-point level. In three-point level, what you do is you define a plane for the image, which is shown right here, and you can click on three places on the image, and then what happens is it tracks a plane corresponding to those 3 points. Now, this image right here is of terraces on a silicon sample. And I'd like to point out that we did this 3-point subtract here, and the image still looks pretty boring. I'm going to come back to this image, and I'm going to show you how using display you can make this image look better. On Gwyddion, to activate this feature, it's right here. It's identified by this red box. So, as I mentioned in my introduction, the next function is displaying the image. Now, displaying will control how the image is visualized, what it looks like when you see it on a computer screen. There's several different display functions. They're 2D and 3D display. You can change palettes, you can do light shading, and you can do histogram adjust. And I'll go through each of these different types of display techniques so that you can see them. The first one is shown right here. On Gwyddion, there's a small cube that's displayed. And I've done this with a test pattern, and you can see now it looks like a 3-dimensional image, and I've changed the color scale to make it look more interesting. And also, from a visualization point of view, you can see this contamination, and now you can see the height of the contamination relative to the features on the surface, so you can see it's quite large. Another display function is palette adjust. On Gwyddion, if you right-click on the color bar that's on the right side of the image, you can select palettes, which are shown right here. There's maybe 100 palettes you can select. And this is a palette that's been applied to that image of not atomic terraces, but terraces on the silicon sample. These are 50 nanometer terraces. And you can see that I have green for one terrace, yellow for the next terrace, and orange for this terrace. And you can see this point defect at the very end, which is very light. Okay, so by choosing a new color palette, I've been able to accentuate this image of terraces. Now, using the computer in a computer modeling, you can artificially create the effect of light shading on the image. That is, a light comes down on the image. It can come down from any angle, and that's shown right here by clicking on this icon on the Gwyddion software for this image. So now what we've done here is we've simulated light coming in from the left side of the image, and you can see that on the left side of the image it's light, on the right side it's dark. Now, this is essentially a derivative technique, and it makes the image look a little bit more like an SEM image. So if I showed you this image without telling you it was an AFM image, you might think it was an SEM image. You can see the defect on the surface right here, and you can see all of the different very small defects on the surface using this technique. So this is a way of accentuating small defects on a surface. So finally, I'm going to talk about histogram adjust, and this is very effective for identifying small features on a flat surface. And so to use histogram adjust, you click on this button right here. And what histogram adjust does is it creates a plot, which is shown down here, of the number of pixels at a certain color scale. So in this image, this small peak right here, it corresponds to these black dots. And this large peak right here corresponds to the top surface. And so what I've done is I've used this histogram adjust feature to highlight just the top features on this surface. You can see now all of the very fine detail of the particles and the surface texture on this sample surface. And so using histogram adjust, you can find very small features that can be in a surface that you wouldn't see if the full color scale was put across the entire height of the surface. There's many kinds of histogram adjust you can do. If you click on this button in Gwyddion, you can select it. It can do it automatically. So at this point, I'm going to stop and ask us if there's any questions associated with display analysis with the Gwyddion software or any software package. We have 2 questions. Go ahead. Will this slideshow be available for later viewing? Yes, it will. And if you write to us at info@afmworkshop.com, we will send you that information. Okay, the next question is inversed. Image line by line nanoparticles. I don't quite understand that question. Is it inversed? I don't think it's inversed. Okay, here's another question. Can we use the software to process images obtained from any AFM instrument? The answer is yes. Gwyddion does support formats from many of the other companies, for example, Bruker or Asylum or any of the other manufacturers. Okay, another question is how to choose the 3 points in the line in the 3-point leveling. You could just do that by clicking on the surface. In fact, Gwyddion has an option that it updates the image in real time. So what you do is you click on the surface with your mouse, and then you can look at the surface and you can move those points around to get the desired effect that you'd like to have. Oh, where the tool is for it? Okay. The tool for 3-point leveling is right here. I'm showing it on the computer screen right now. Okay, it's right there. You just click on that and it brings up a box, and then you can click on the screen. All right, so thank you for those questions. I'm going to move on to the third topic, which is analyzing the images. Now, atomic force microscope images are three-dimensional arrays of numbers, and so therefore there's many types of analysis you can do. I've listed here 4 types: line analysis, surface analysis, probe deconstruction or deconvolution, grain analysis. This is also particle analysis, metrology analysis. And again, many of the software packages you purchase have these kinds of analysis. Today I'm going to talk about line analysis and surface roughness. Line analysis, line profile is the most common type of analysis done on AFM images. It allows you to find the distance between surface features, the height of surface features, and the width of surface features. So on Gwyddion, you click on this button right here, and that allows you to draw a line on a surface across the feature you want to look at. A window comes up, and if you push apply on that window, you'll get this window right here. And then what you can do is click on this window, and this brings up these cursors. And you can move these cursors around on the image, and this allows you to measure the height and width of a feature. So in this case right here, the cursor's almost at the top of this nanoparticle, and this cursor's at the bottom. And so this allows us to see that this nanoparticle is 14.78 nanometers high. You can do multiple lines with Gwyddion, or most of the software packages. That is, you can go from here to here. You can do multiple particles or surface features and get the distance between the features or the width of the features. Now, always remember that when you do the width of a feature with an atomic force microscope, it's going to be a combination of the tip width with the width of the actual sample you're looking at. The next most common analysis technique is surface texture. And on Gwyddion software, surface texture is activated with this button right here. You can see a bunch of symbols. If you click on this, this window will come up. And Gwyddion gives you the capability of including or excluding areas of the surface. So in this case right here, what I've done is I've drawn a box around this part of the surface image, and I've put include. So this list of parameters right here is associated with only with this area right here. So you can see that the surface roughness, RA or SA, is 3.7 nanometers for this area. Now I could easily do exclude and just do all the part outside of this. This is very important for measuring the noise floor of your instrument, which is very critical to the operation of atomic force microscope. So the conclusion, and don't go away yet because I'm going to give away a free book. The conclusion is that you can process images, which is associated with changing the numbers in the matrix. You can display images, which has to do with how the images are, the matrix is viewed. You can do analysis. It'll take you about an hour or two to learn how to use Gwyddion software at a basic level, which I've presented here. If you want to do the more advanced functions, you might spend as much as a month to learn all of the details of the image processing to become an expert in this. And we really recommend, if you're going to use your AFM a lot, to invest the time to learn all the functions. Okay, do we have any questions? Okay, it says image contrast inverse in the nanoparticle image. Let me look into this. I guess maybe the color scale looked backwards on that, where maybe the particles looked like they were going into the surface. So we'll go back and review that after we do the presentation. Okay, and the next question is, how do I choose which 3 points I should choose? And I think we answered that already. But when you bring that up on the Gwyddion software, you can put your mouse and click on the different points. And you can update the image in real time. And by doing so, you visually will see which ones to use. Okay, there's some more questions. I'm very new to AFM, so I apologize if this question has a very obvious answer. What is the purpose of three-point leveling? What information is retrieved? Well, three-point leveling is a good technique for using for specific kinds of samples that have multiple levels in it. Quite often, samples like terraces, atomic terraces, or images of semiconductor device structures with many levels, they don't level well with planar polynomial leveling or line leveling. So three-point level can be very good for those kinds of images. Okay, how to use two lateral force images, trace and retrace, to make a friction image with Gwyddion? That's a good question, and I don't have an answer. My suggestion would be to go onto the Gwyddion website and see if there's a way of overlaying the images, and that would probably be the best way to do it. Any more questions? Okay, one more question. Can Gwyddion be used on TIFF and JPEG images? I believe it can. On Gwyddion, if you go into the file import, it has a lot of options, and I believe you can import those types of images. Okay, how to get a 3D image with Gwyddion. On Gwyddion, on the control panel, you'll see a picture of a cube. It's sort of a third of the way down the right-hand side. If you click on that cube, you'll get a 3D image. I have an idea that I just thought of. If some of you would like to email us at info@afmworkshop.com, we can send you some images and tell you where to get this presentation, and you can use it to sort of work your way through Gwyddion. So we can offer that to anybody that would like to contact us. Okay, so I hope this webinar has been beneficial to you. Any feedback you can give us to help us improve it, we'd like to have. And again, our company is AFM Workshop, and you can see our website at afmworkshop.com. Thank you very much.